Interim Dean Nick Pace here, and I am with Professor Sathish Natarajan's lab. We're here in the lab with excellent students, and we have a whole lot to learn. So, Professor, tell us what happens here.
Thanks so much for joining us. And I think we are interested in nutrition research, which dietary nutrient that could help prevent maternal fetal complications. So we have a project going on, how fatty acid oxidation could be affected and a fetus could be affected. It's an acute fatty liver pregnancy. That's how I started my lab. And then the branches all happen after that.
So show me around. What can I learn?
So firstly, I'm going to show you the Zika virus group. Basically, we have three groups. One is just the normal group, which is not infected. And the second group is the Zika virus group, which technically it is infected with the virus.
Okay.
And the third group, we infected the virus and also we gave the macadamia, the palmitoleate diet.
And I'll be able to see big differences.
Yes, yeah. If the brain is infected with the Zika virus, then it shows green in color.
Okay, the bright.
Yeah, the bright colors. So these are basically the nuclei, nucleus of the cell. The virus, if the previous image I showed you, it's just the virus alone. Okay, so we have to say that the virus is the one which infects the cells, right? So we take an image, we click this as an image, and we also take with the same panel, we go to the green signal, which is the virus signal, and we merge that.
Okay.
So that we say that, yes, these cells are infected with this virus. So this is how we represent the image if we want to publish in a journal, in a scientific journals, because we say these are the cells, blue colors are the cells, and the green colors are the virus. So the cells are infected with the virus. I like this so much because we can see here is the virus, here is the cell. So I like this technique so much. Yeah.
And it is easier to do this volume. When we go for lesser volumes, we need to be more careful that we are taking the correct amount and then we are not causing any bubbles or if it is not. Do you want to try it out?
Sure. Sathish, are you hiring in the lab currently?
Absolutely. If the experiment works really well, then we would probably be in your office.
Okay.
Do one level of pipette as a custom.
It's a very basic first test. Okay, so same process to fill the whole tray.
Yeah.
Okay.
And then we keep it in a carbon dioxide free incubator for 4 to 48 hours to the maximum. And then tomorrow what we do is we remove this and then we keep cells. So we seed cells in a similar plate. And tomorrow what we do is, instead of this hydrating medium, we will put this cartilage into the cells and we'll place it in a seahorse mission where it reads the value of how much oxygen is the cells being consumed. And based on that, we will know how active the cells are and how much cells are there. So it's basically testing the mitochondrial function. So the mitochondria is source of oxygen. So when you measure the oxygen function in it, you will know how mitochondria is functional or dysfunctional in the particular cell group. So if I'm doing a treatment with some fatty acid, it might block the mitochondrial function, actual mitochondrial function of the cells, which will result in a decrease in the oxygen consumption that the cells use for their activity.
I'm interested in hypertensive disorders of pregnancy. So this is women who come to the clinic and they present with hypertension during their pregnancy. So this is like gestational hypertension, chronic hypertension, preeclampsia.
And so that could be caused by pregnancy. They might be women that did not have hypertension before they were pregnant.
Yes. So pregnancy is a cardiovascular stress test alone.
Okay.
So I look at that, and this is common in about 10% of pregnancies worldwide. So the one thing we look at is nutrition here. That is one thing we all have in common. And I'm interested in omega-3 fatty acids. So there's a specific lipid metabolite that I study that is actually showing improvement in migration of these trophoblast cells to be able to migrate into the uterus of the maternal side to help again with the exchange. So essentially is we can take the proteins and we can basically embed them into a gel and then put them on a membrane. And they're separated based on the size. And this is a membrane, okay, and it has proteins on it. And we can take our proteins of interest and we can actually have them bind to regions on the protein so we can actually visually see, semi-quantitatively, see how much protein is there because these proteins are due actions. So these are all different subjects here. And we're actually looking at this protein band, this bandwidth here, this band's up here. Okay, so these are normal tensive women, okay, normal. And then we see a decrease in these proteins up here in our hypertensive samples. It's very little, but it is there. So it's showing that there is an actually kind of like opposite what you think, but there is an increase in oxidative stress in these women and the metabolism isn't working overall.
So you are much more concerned with this.
Yes, I'm concerned with the hypertensive subject because they're not producing enough of this protein that is needed to help mitigate an oxidative response.
I'm working on fibroblasts now. Okay, so if I'm planning on some experiment or something, a week or 2 weeks before, I will take cells from the liquid nitrogen. So they immediately, I cannot work on the experiment. They need some time to maintain. So after that, I will start my experiment. Okay, it's like a step-by-step process. So in the top box, we have the AC16 cells. And then in the bottom, we will be having the fibroblast cells. So in each vial, you will be having the cell line name and then the passage number. Culturing is like multiplying the cells so that every cells will be having a viability range. Since I'm working on the fatty acid oxidation disorder and then to see the mitochondrial dysfunction, I will be working with the fibroblast cells. That means like the control fibroblast patient cells as well as the HADHA gene mutant cells.
And what are you hoping to see?
So each cell will be having a different cell structure. Okay, so I know like how the fibroblast cells will be. They will be like elongated cells. It's always good to start with the cells and then you will be having a data and then you will be having an idea.
And you can build
we can build from there
safely. I'm really excited to see what's around the corner for all your discoveries. Thank you for letting me look around and for educating me. This is wonderful.